DNA is the instruction manual for life on Earth. Often referred to as the world's biggest codebook, it encodes the fundamental properties of an organism and is passed down from cell to cell, from generation to generation. Our DNA codebook contains words made up of only 4 basic letters: A, C, G, and T. The DNA double helix in humans consists of 3 billion specific arrangements of these letters. Arranged correctly, they provide the building instructions for proteins and other important molecules for cellular function. Although DNA usually replicates with fairly high fidelity, mistakes do happen and changes in the DNA sequence can occur. These genetic alterations can be inherited and run in families, or are newly acquired and unexpected. Sometimes these changes are harmless, but sometimes they alter the functionality of a cell and can be the root cause of one of the thousands of rare, monogenetic, and sometimes life-threatening diseases. Gene editing is the targeted manipulation of genetic material. You can think of it like a Swiss Army knife. It can perform many functions, such as inserting functional genes into cells or editing mutations in the genome. It offers a diversity of ways to edit DNA, enabling a wide range of therapeutic applications. Let me introduce you to gene editing and why it is an essential platform component for Bayer's CGT strategy. Gene editing treats diseases at the genetic level by directly modifying a patient's DNA. It often involves the creation of targeted breaks in the DNA with or without instructions for repair. Newer types of gene editing technologies encourage editing without the need for a double-strand break. And regardless of the exact mechanism of choice, therapeutic gene editing can be achieved via a number of different techniques. First, genetic disruption or inactivation. Often mutations in genes can result in the expression of a mutated toxic form of protein. A gene editing approach would be to inactivate the mutated gene in order to remove all toxic protein expression. By tackling the underlying root cause of the disease, the hope is to relieve the patient of all symptoms and treat the disease in its entirety, as opposed to treating 1 or maybe 2 symptoms. Another technique is gene correction. In this technique, an additional piece of DNA is provided and used to repair a gene. Rather like the find and replace function on a computer, this technique aims to fix small mutations in faulty genes and can be used to correct the single point mutation in 3 billion letters that causes disease. Gene insertion, on the other hand, can be used to add entirely new genes into cells, providing new functionality and therapeutic benefit to the patient. And genetic deletion can be used to remove faulty or unwanted pieces of DNA from the genome. An example here would be to remove large repeat regions from a gene that cause a non-functional or toxic protein. Research is ongoing to create gene editing therapies that can combat cancer, blood diseases, infectious diseases, and rare inherited genetic diseases. The beauty of gene editing is that it offers a diversity of different ways to manipulate DNA. By understanding the underlying genetics that cause disease, the right gene editing technique can be coupled to the right disease. There are different gene editing technologies that can be used to perform these different gene editing techniques. You may have heard about a specific gene editing technology called CRISPR, an acronym for the Clustered Regularly Interspaced Short Palindromic Repeats system found in bacteria and archaea. CRISPR-Cas is the most versatile gene editing technology, and while being the youngest, it is superior to its predecessors as it combines efficacy and flexibility. The CRISPR system consists of 2 parts. First is the Cas enzyme itself, and the second is the guide RNA, the tool used to locate the target DNA sequence. Cas proteins, most commonly Cas9, are a specific class of enzymes that break the target DNA sequence. These proteins work with the guide RNA, which is engineered with a specific sequence that tells the Cas protein exactly where to cut the DNA. Once the DNA is cut, the endogenous methods of DNA repair in cells is hijacked to bring about the desired therapeutic editing outcome. This break can be repaired with or without instructions on how to do so, thus leading to different end results. Gene editing can therefore be used to correct disease-causing point mutations, to precisely add or delete genes, to add new functionality, or even to increase or decrease gene expression. Gene editing as a therapeutic treatment for genetic disease can be applied inside and outside the patient to treat a variety of diseases and provide diverse clinical benefits. Ex vivo gene editing edits genes in cells outside of the patient, most commonly stem cells or immune cells. In vivo gene editing, on the other hand, edits genes in cells inside the patient. To deliver the gene editing components to the right tissues and organs, they are packaged into delivery vehicles such as viruses or lipid nanoparticles. And many of these delivery vehicles are already FDA approved for non-gene editing purposes. Gene editing can also serve as an enabling technology for cell therapies. It can be used to alter and therefore improve therapeutic cells, thereby enabling next-generation cell therapies with enhanced functionalities. For example, increased potency and persistence, reduced immunogenicity, or inclusion of safeguarding mechanisms, all of which can improve the safety and the efficacy of cell therapies or enable a larger patient pool for treatment. While gene editing-based therapeutics are not currently on the market, the number of clinical trials are increasing year on year, with promising early results already being published for both the ex vivo and the in vivo use cases. Furthermore, the pace of technological innovation for gene editing is truly incredible. Researchers are tweaking the current technologies to improve things like fidelity and efficacy, and new types of technologies are being invented from academic labs all around the world. While the field as a whole is still relatively immature, the speed of innovation means novel therapies are advancing at rapid speed towards the clinic. Because our gene editing platform serves a dual role, driving standalone therapeutic applications and enabling next-generation cell therapies, gene editing is an essential platform component for the future success of all of Bayer's CGT strategy.